Okay, welcome again back to Engineering 443, 543. We're in Lecture 7. We're going to be talking about chemical fields, diffusion, decay, and secretion uptake. I need to remind you that this lecture is live streamed on YouTube and is recorded. So please remember that during the class. So I want to start out by doing some of the student presentations that we've been deferring. And then we will come back and go through chemical fields a little bit more. That's a very rich subject. One could spend almost a whole class talking about chemical reactions, chemical fields, and so on. But we're going to go through it fairly quickly. Today we'll be focusing on some of the basics and getting people up to speed on that. So some people I know said that they were ready to do their miniature presentations on their projects. And so if anybody is ready to present today, why don't we turn the floor over to them for the moment. Connor, I think you said you wanted to do that.
Yeah, I think we're good. Andrew, are you good to go? Okay, awesome. In that case, I can share screen and we can just do what we did last time, Andrew, and you can tell me when to click through.
I thought I was gonna.
Oh, you can click through if you want. I'm not opposed either way. Go for it.
Here we go. And how do we turn this full screen? Is there a specific button to turn this full screen?
Yeah, go to slideshow right next to share in the upper right corner. Further to the, yeah. Further. Like go way to the right, like all the way.
Okay.
No, that's left. Okay. Yeah, right there. All right, we got it. Okay, so this is going to be a slightly more in-depth introduction to the very short PowerPoint presentation that we gave the other week about kind of like trying to look at transition cases to multicellularity. Next slide. All right, so first, a quick overview. Generally, when it comes to some of the theoretical literature around the transition from unicellularity to multicellularity, there's a lot of assumptions baked into it. So for example, in simulations, usually the fitness functions will explicitly encourage multicellular conglomerates to form, or assumptions will be made about the benefits of multicellularity. So we don't actually have a ton of information about the types of conditions that lead to multicellularity, and also in terms of the types of functions that we can get out of it on more of an emergent basis. And this is a little bit problematic. So I posted a picture of a book here that someone told me was deeply influential for them for thinking about multicellularity. And one of the things that gets discussed pretty early on is that there's kind of a contradiction in place when you have cellular differentiation and only some cells get to reproduce, because this goes against the interest of the singular cells as individuals when there's a germline and soma barrier. But there's some interesting kind of early cases. So next slide, please. So here I just wanted to go ahead and post a couple of cases. They're actually a little bit further along than the one that we're going to talk about. So on the left here, I have Volvox, a type of green algae, and this is specifically looking at its asexual reproductive cycle. So what you can see here is that you can see that there's hatching, maturation, cleavage, and inversion. And the really key thing that I want you to pay attention to is that there's differentiation. So at the end, those kind of pockets inside will come out as new organisms, and the rest of the soma will senescence and die. And this is similar to what we see in slime molds when there's reproduction from the stalk. So the part of the reason I'm showing this is because these are some like cases that have been really transformative for how people think about the transition to multicellularity. And the simulation that we're going to talk about actually goes back a step further than this because it does not get to the stage of cellular differentiation. Next slide, please. All right, so I'm going to give a really quick kind of like bird's eye view overview of the paper. You've seen this before, and then Andrew is going to go into more depth for a little bit. So there's a few elements that are going to come up. One is adhesion between cells, and this is going to be subject to evolution. So how much the cells stick together based off of a string of bits that determine the binding strength. And there's also a similar bit length for the environment, which I thought was a weird choice, but we can talk about that, I suppose. There's also persistent migration, where the cells would kind of continue along the path that they were following before, as opposed to just chemotexing straight. And a really important difference compared to the simulations we've seen is that there's a chemical gradient with holes in it. So it's probably hard to see here, but if you zoomed in really, really close, you would see that there's a bunch of white specks in this chemical field. And the result of that is that it's harder to kind of climb the gradient. And then that allows us to have season length. So what ends up happening is that at a certain number of time steps, the source of that chemical gradient is going to move. Cells are going to reproduce based off of how far they made it up the gradient. A certain subset of those cells will be selected at random to be culled, and then they will climb the gradient again. And the idea is to kind of track the either waning or growth of the adhesion between cells as multicellular conglomerates. So down here, we see that process. There's a clump climbing the gradient. Once the season changes, they reproduce, a certain number of them get culled, so there's always a constant 200 cells, and then they go to find the new location once again, and that's going to be the basic structure of the simulation. Now it's all you, Andrew.
Alrighty. So yeah, I'm just gonna go into more detail about these figures. Here we have two different, there's the receptor and the ligand.
Sorry, your sound isn't working.
I think we can hear you, but it's just a little bit faint, Andrew. So you might just have to be boisterous about it.
Is this getting any better?
A little bit staticky, but you're definitely loud.
Oh no. Let me see if I can.
You're actually excellent right now compared to before.
Okay. All right. Okay. So there's a receptor and there's a ligand, and each of them have their own bit strings associated with them. And if their strength is determined by how complementary one is to the other. And
Andrew, I'm so sorry to do this to you, but you got quiet again. I'm really not trying to interrupt your flow.
Maybe automatically change it. Is that any better, or is that too loud?
It's not that it's too loud. It's that there's a pretty. I mean, maybe it's just me, but there's a pretty aggressive static that is coming through with that for me.
It's like a background noise.
Yeah,
probably.
What?
Not to propose a non-technological solution, but maybe just speak loudly and close to the microphone on the level that doesn't.
I think I know what it is. I'm gonna have to take a moment to adjust my mic though.
Okay. Would you like? I guess. Do you want me to keep going and we can just switch things up, or? Okay.
No, it should take a few seconds.
Professor Glazier, these I think one of these is the books that you mentioned during the last meeting. I forgot the title. I wasn't able to find it on the Canvas page, so thank you for sharing it here.
Some of them you showed before, I think, and I know you have them, but I thought some of them might be of interest to other people in the class. So I thought I'd add those links.
I mean, I'll never complain about having too many books, so.
Well, so the major transitions of evolution is a classic. as I say, it's controversial. People have argued that everything, but it's certainly for a book that to excite a lot of discussion and thought, it's important. And I'm not saying it's wrong. I'm just saying that there's been everything in it has been argued over. And then the two papers you mentioned, Stan, you showed Stan Murray's culmination work, but Stan's earlier work was on the collective migration simulations. And so I put the two papers that he did on collective migration in the chat as well.
Is my audio any better now? Has it
Class,
gotten better?
it is not.
Oh, no. All right, well.
Just
yeah,
speak enthusiastically and loudly.
As long as I'm just concerned about the static, like that's.
No, there's no static right now, and we can hear you when you have a lot of inflection. So I would say just roll with it from there, and we'll make do.
Okay. All right, so refocusing back into the receptors and ligands. So the more complementary the bit strings are, the stronger it is, and when you have a strong, when they're very complementary, they are strongly binding. And the strict description of the environment we have over here, there's a focal point, and going from the focal point out into the perimeters, it decreases in the likelihood of, or it decreases in the intensity, whereas our model is going to be a little bit different. Here we have it decreasing in intensity, but also getting more patchy the further away from the gradient you go from. The further away the pixel is from the focal point. And then there's also the chemotaxis, where at least in this paper, what they calculate is they calculate the center of mass of the gradient within the cell. So over here, even though the chemical is actually has less is less intense than over here, because there is more points, the center masses will be here, and the angle of chemotaxis ends up going in that direction. And that is combined with the persistent migration. So there is, if it's already going, if the cell is already moving in one direction, it will continue going in that direction, but it's getting pulled by the force of the chemotaxis. So that's what's being shown here, where this is the angle of persistent migration, whereas the chemotaxis event is actually pulling it down this direction to the point where the persistent migration and the chemotaxis get closely aligned over time. And now it's up to now it's Connor.
This was actually still you, but I can also do this if you'd like.
Sure.
Okay, so general results here. So I'm not going to go into all these plots in a ton of detail, but I'm going to point out a few key things. So first of all, on plot A, gamma here, what we see here is the adhesion strength, which they are calculating by taking it was contact energy of the cell and medium and minus the cell and cell divided by 2. And what we end up seeing is that one cell is that plot there in yellow. And then as the adhesion starts increasing, the center of mass relative to the kind of the focal point of that chemical field starts. It's able to traverse that distance quicker when they're in a multicellular conglomerate. And most of the rest of these plots are getting into that in more detail. So B is looking at the cell closest to the gradient peak. C is the mean squared distance, where gamma equals 6. And the yellow one is one cell. And something that's really interesting, you look at these two images on the right, is that here on the top, we see the path of one cell, and it's pretty scattered. Part of that is that the single cells have a really hard time dealing with the noisy gradient, and they also are struggling because of the persistent migration. But on the bottom one, in the conglomerate, it's not that the cells are now all of a sudden following very linear paths, but because we can still see the trajectories of individual cells being traced there, the red and the yellow one. But ultimately, they are, as a whole, following a more linear path. And part of that is that through the adhesion, they are constraining momentum together, and they also have a larger surface area in terms of picking up the chemical gradient. Next slide, please. Okay, so this is one of the interesting results that they found here, and that's that you can get either unicellular strategies or multicellular conglomerate strategies based off of the season length. So remember how I said that the source of the chemical field was changing at different time intervals? So if we look at figure B here, the bottom one, we're seeing on the x-axis the duration of season lengths. And what's happening is that we're getting an evolutionary bistability where the season length is kind of at that 20 times 10 to the third Monte Carlo step range. That's what we're seeing in the left image with all the single cells. They are evolving to have less adhesion together. And the reason for that is that they're kind of stumbling along and their position is not reset between seasons. So there's a chance that they can stumble upon the new chemical focal point that's going to emerge. And the season is changing too fast for the multicellular conglomerate to actually get there and have a reasonable chance at reproducing. This is compared to the longer season length, in which that's a viable strategy. Next slide, please. So we don't need to watch this full video, but this was a case of that process taking place. So it starts with neutral adherence. So we're seeing over time, this is the range 100 times 10 to the third where we would expect to see a multicellular strategy emerge, and that's what we're eventually getting over time. The peak of the gradient is moving, cells find it, they reproduce according to how close they get, and there's also a mutation rate for the ligand binding sites, and over time we're seeing that they're becoming more and more adhesive relative to each other. And that just continues, so we can go on from there. Okay. So general reflections on the paper, it's very, very well documented, but there are some odd design choices about it. One of which is that the chemotaxis was very particular, but we talked to Roland and he was saying that a more general implementation would be fine. Another thing that I at least found odd about it was that they represented contact energy with the medium, having its own kind of bit length, similar to the ligand binding sites. Still kind of trying to think about how necessary that is to demonstrate the general effect there. But there's some interesting implications there for talking about the transition to multicellularity, because like I said, it doesn't get into the stage of kind of like presumed cellular differentiation in terms of reproductive and non-reproductive cell types. So this is kind of an earlier case study. And it sets up a good foundation for a qualitative model. So one of the things that we're trying to do right now is that we want to reproduce some of the general results and then think about an extension that could be made, because I think that all of us are on the same page that there's plenty of room for that. Next slide, please. And yeah, so now I believe, Andrew, this is you again. You were, yeah.
So now this is kind of going over what we had this in our previous presentation a few weeks ago, but just to reiterate over it, that they're using the cellular pots model where the cells are moving stochastically and all their actions are determined based off of minimizing energy. There is cell migration, where this is the persistent migration where they consistently walk in a single direction, and then so they have a force, essentially a force that's being applied to keep them moving in a specific direction, and that changes over time, and that's what's happening with when we're combining this persistent migration with the chemotaxis. And then cell replication is when cells are dividing after the season is over, and they divide along its short axis. And cell death, where after the replication, half of the cells are culled, and then those are being used for the next season. And our paper is going to be implementing chemotaxis differently, where so they are they're taking the average level of chemotaxis underneath the cell, whereas we're going to be using the plugin for chemotaxis in CompuCell, which is calculated by minimizing energy. And their total fitness each individual cell's fitness is calculated by its distance from the peak of the gradient. And what is being mutated is the binding term between individual cells, whereas instead of a bit string, it's going to be an integer from 1 to 6, and the lowest number of the two between the two cells indicates the binding strength. And then after a certain amount of Monte Carlo steps, the season is over, and then we calculate the fitness where the highest cells, or the cells with the highest fitness, have a chance to reproduce. And then that is our presentation.
So Joel, go ahead.
I think Joel was just clapping.
I was clapping,
Virtually.
but yes, it's very interesting, all the paper. One of the questions that I have, this thing of like you deciding to connect using a integer, is that like for the whole cell, or is there specific size of the cell that you're thinking? Is there a way of doing that?
So it's the whole cell, and it's determined by basically like also there was a mistype that I forgot to catch in the slide, which is that it ranges 0 to 6. So the idea is that you take it's the lowest number that matches basically, right? So if it was 5 and 4, then the binding strength between the cells would be 4. If it's 0 and 6, then the binding strength would be 0. And then that's just considered to be the interaction between the cells. It's not geometrically specific.
So I mean, this is already plenty if you can get it done in the semester. But there are a couple of extensions that would be very classic major transitions of evolution extensions that you alluded to a little bit. One of them would be one of the classic, although it's not completely true, distinctions between germ cells and somatic cells. Because in general, we think of germ cells as not being able to do so much. Stem cells generally don't do that much, except to be able to reproduce. Now, of course, there's some pretty big exceptions to that, like sperm that swim pretty well. But they also have short lifetimes because developed sperm has very small cytoplasm, so it can't last very long. So one possibility would be to do a simulation in which you have some idea of germline somatic separation, where the germline cells are non-motile, they have to be carried by everybody else. And the somatic cells then do the chemotaxis. And then there's a heritable number, which is the probability of the cells being germ-like or somatic. And then you would typically find that if it was hard for the cells to move to a favorable location, you'd need more somatic cells per germ cell to be able to get your organism to survive. So that's one hypothesis about germline somatic separation. Another one is the buffering hypothesis that shows up in Volvox, where you say that the probability of survival of your germ cells is higher if you're in a bigger cluster. Not in general, because then you're giving the answer. But if the hostile environment kills off the cells at the boundary of the aggregate, but not in the middle, then there's some probability that the bigger the aggregate is, the more likely you are not to get wiped out. And so that's another way you can have the environment then select germline somatic separation. And so if you happen to finish the project you already have, those could be extensions possibly of this idea.
Doing ambulance start driving by right as I unmuted. Hopefully everyone's all right. So I guess my general question, we could talk more about this when it's not taking up class time, but it would be that one of the things I thought was really beautiful about this simulation that Andrew and I were discussing a lot was kind of like the lack of assumptions compared to other models. So in the case of having like germ cells that need to be carried, I guess I'm trying to figure out how to reach those points where those strategies could be investigated without actually presupposing that there's already, I think we talked very briefly once about the possibility of making it so cellular differentiation could happen gradually in this style simulation, which might be one possible way to get at it, but I'm a little bit fuzzy on the details for implementation there. So that would probably be a longer conversation.
So yeah, we could talk. I'll just make one general comment, which is not universal because biology always has counterexamples. But already in bacteria, many species of bacteria have what are called the swimmer phase and a vegetative phase. And also many eukaryotic cells also have the same thing, which is that for cell division in eukaryotes, you need the microtubules and the actin to be recruited to do the cell division. It was sort of a truism in the textbooks that cells couldn't migrate and divide at the same time. And that's not quite true. But to migrate effectively and divide at the same time is not possible because essentially the same machinery is being repurposed. And so you can imagine a cell that divides with a high rate is not very good at migrating. And so then you can have a trade-off between replication rate and migratory ability. And you can imagine in many things Pareto optimals constraints lead you to pick either the full replication pathway or the optimum is actually of the extremes rather than in some combination. Now, whether that's true or whether the optimum is in the middle is something you have to discover. So I think it's possible to think about that, but yes, it's difficult. Serendipity is very hard. In biology, you can have evolution generate completely new mechanisms that didn't exist before. The problem when you're doing computing is you have to put in by hand at some level the capability to have those mechanisms. The organization of your cells into a multicellular organism, the multicellular organism could do things that the individual cells can't, even in the simulation. But the individual cells, to some extent, you have to predefine what they're capable of doing. And so that does make some of these evolutionary questions a little tricky to do without prejudging the outcome. It's very easy when you do computational evolutionary models to basically program the outcome in from the beginning. And so I appreciate your desire to avoid doing that. And so I'm happy to talk about that more. And our biologists in the room are welcome to weigh in on that particular issue because it is a tricky one. Okay, does anybody else want to present any more questions for our speakers, and if before we move on? Okay. Is anybody else ready to present today? Josh?
Yeah, I'm ready to present if that's okay.
Okay, please go ahead.
All right, just one second. Okay, Can everyone see that?
Yep, we've got you.
Yep, looks good.
Okay, very quickly, I will put in the chat, this is the link to the paper if you would like to follow along. All right, so the paper that I'm basing my project off of, it is called Emergent Networks, a Slime Mold Simulation. It was written by an undergraduate, I believe, for his bachelor's thesis. His name is Nicholas Simler. I was able to get in contact with him. He sent me all the code for it. So that's a pretty good start. However, this was written in 2011. So I think I mentioned last class that it'll probably take some work to port that over to CompuCell 4.x, but hopefully it's not too hard. So now just getting into the paper. I just go through every section that Nicholas has. If you've gotten a chance to look at it, it's very short. It's only 20 pages, but over 10 of them are references and figures. And each section is, I think the longest one is 5 paragraphs. So it's not very detailed. I think that might be a part of my project, is to go into some of the more technical aspects. So for example, in the introduction, he just briefly discusses the nature and history of computational biology, mentions things about like Turing patterns, how one of the last thing Alan Turing did was try and model biological behavior, kind of setting the scene. Then he starts talking about this Physarum polycephalum, which is the specific type of slime mold that he simulated. But specifically, the polycephalum has a lot of different stages, but more specifically, he took a look at the plasmodium stage, which the slime mold, that's where it ventures out into the environment to create these networks to find the food sources. And then he briefly talks about some applications and stuff like that. For example, he compares a lot of the slime mold networks built to like complex human-made systems like railways and stuff like that. So next, he goes into more detail about the slime mold. So in this plasmodium stage where it's exploring its environment, it's an amoeba-like organism with a broad tubular network. And specifically, the slime mold performs a computation to create the networks to communicate, whether it, the path that it found leads to a food source or it leads to an obstacle or a boundary. So this computation is constrained by the environment. While it's exploring, it has this 2-part system. It's a tubular network, so it's constantly, as it's exploring, it creates these actin myosin fibers, and it transports chemicals and food throughout these to communicate the direction and whether it's heading in an optimal direction or not. So the flow of plasmodium steers the fibers being created, and the direction of the fibers limits the flow of the plasmodium. And so as it's creating these tubular networks, there's some developmental rules. So namely, the more food a fiber or a tube gets, the more the thicker it'll be. So essentially long, thin, and less connected tubes are essentially the tubes that don't lead to any food. So it will try and minimize those. However, the tube size does have an upper bound. I think this is more due to computational resources. And well, I guess I don't totally understand the biology, but I think there is some biological reasons for the upper bound as well. And naturally, the plasmodium flow in the tube increases with the size of the tube. More food can flow and more signaling chemicals. So after going through kind of the biological process of the slime mold, he starts going into the framework that he used to model it. He briefly talks about why he briefly introduces the CPM model, explaining its relations to the Ising model, which is a very popular one. He explains why he chose CompuCell, which is essentially that it was the most robust library at the time. And then he very briefly goes over some of, sorry about that. He briefly goes over some of the main ideas, because it's mainly a chemical field that we're simulating. So he goes over the chemical field will utilize secretion, diffusion, and decay. So I believe today's lecture will be very relevant for this. So now he goes into the model. The simulation is grounded in 3 concepts: direction, movement, and fitness. So it's kind of, you give the slime mold an environment, place it anywhere you want, and then you also place food and obstacles or repellents. So you have to define attractive and repulsive chemical fields. As far as the movement goes, the movement is driven by an autocatalytic loop. And this autocatalytic loop kind of makes sure that it goes from a blob to an actual tubular structure. He talks about some of the contact strengths. So low cohesion, secretion of attractant scalar with restricted volume. So this kind of makes sure that it's moving in the right direction. And so with just direction and movement, that the slime mold just explores the environment pretty much randomly, but to actually get a optimized, structure to find the path of least distance to a food source, you actually have to define some kind of fitness to get this optimal structure. And this is done through growth and oscillation to increase and decrease the volume, which is talked about more here. So the growth blob formation is penalized, and as it oscillates the chemical signal, it closes the small tube loops. So it kind of, going back to the rules for tube formation, that kind of creates that. And then, so these are just more in detail about the attraction, repulsion. Naturally, the plasmodium is attracted to food sources, repulsed by unfavorable conditions. I was reading in some different papers that slime molds they're semi-intelligent in the sense that they can actually, I believe the researchers, they had an actual slime mold explore an environment, and then they would blow cold air at fixed intervals, and then they would stop, but the slime mold would keep, whenever the cold air filled the petri dish, it would kind of slow the growth. And even after when they didn't flash the cold air, it still slowed the growth at the set intervals when they were doing it. So they do have other forms of learning like that. But in this, it's essentially, we have to create all of that in the form of toxins and repulses. And then there are some minor changes just due to the fact that it's not a real biological system. So for example, all chemicals saturate at a specified level. This is in order to mirror the finite molecular size. And food sources increase attractor secretion at fixed intervals, so they appear less homogeneous. And so, like I said, he didn't really go too much into detail about what the results mean. He was more concerned with just getting, I think, the simulation to work. But it seems to have been successfully implemented. So he talks about, in literature, real slime molds have been able to recreate minimally spanning trees. And so he tested that And I don't know how familiar everyone is with minimal spanning trees, but this is one with 3 nodes. So that's pretty promising. And then on the right, this is a more complex food system. And there's 3 layers. So this first one is just the normal cell field. This, I believe, is the attractive and repulsive field. So as you can see, the concentration is really high here. And this is just the barrier. And so I think my main goals for the project are going to be to kind of go into more detail about the more advanced topics of the modeling and things like that, and then of course porting it to NanoHub, and then maybe making it interactive, so you could just drag and drop food sources and draw barriers and other things like that. And that's all I have.
Questions, comments? I put a few links to some relevant papers in the chat.
Okay.
This is a topic that's been it's highly topical, still definitely under research. My old lab mate when we were graduate students, Marcelo Magnasco, who's now at Rockefeller University, has written fairly extensively on slime molds and transport. And so you might see if any of the papers that I've put in here are useful. Because you're dealing with an older paper, these are slightly newer references that might you're certainly not. I mean, your assignment is to replicate the paper that you picked, but you may find that with relatively little additional work, you could take the work forward to what's happened in the past couple of years. And so some of those might be of interest to people, but that's great.
Yeah, thank you.
My only comment is on the porting over of old CompiCell code to new CompiCell. What I usually recommend doing is to start a new project and copy block of code by block of code, not too many lines at a time, because too many functions change names and so forth. And reading and comprehending somebody else's code is already an endeavor. And then having to translate that code into new software is a lot more on top of that.
Yeah. I just briefly tried to open the .ccd in the player, and it threw a bunch of warnings. So I was planning on just creating a new project and porting it over. But I will say the nice thing is that his comments are fairly more informative than the paper. So it might be, yeah.
That's not common.
So.
I mean, as a practical matter, what you want to do is identify the plugins that he uses and the steppables that he uses, and then use Wizard to build that structure. And then copy the code block by block into that framework. Because the main things that have changed, there are some names of things that have changed, but the main things that have changed are the boilerplate code at the beginning of the steppables and things like this. And so if you use the Twidit wizard to create the structure, it'll give you all the boilerplate code. But then, say, looping over cells hasn't changed. So most of the logic of the Python won't have changed. And then we could definitely sit with you. So probably I'd ask both Connor and Josh, I'd ask you to make one-on-one appointments with Giuliano, and ideally this week or early next week, and try to sit down and start spend an hour and a half trying to power through these things, so that
definitely
we get stuck, I can I'm happy to get involved, but Giuliano probably is actually a better troubleshooter than I am. Okay.
All right.
Lovely. Anybody else want to present today? Okay, so the people who didn't present already have to present next week. It's definitive. I know Joel and Jackson, you've changed topics, so maybe you need an extra week to get together. Don't have to be total. You don't have to be completely ready. Just have to have something to present. Okay. Anything else before we get back to the lecture qua lecture? Okay, thank you everyone for the presentations. I appreciate it. I'd like to come back and just briefly review some of the ideas that we were working with last time. Some of these came up a little bit in the homework that I graded, and I tried to make some comments about this. We did a couple of in-class exercises. One was cell division. This is actually when we skipped, so in a sense you have to do it on your own. A cell division where you have a single cell that's proliferating, a stem-like cell, and a terminally differentiated cells. And that difference between this and the examples we did in the homework was that in this case you get essentially linear growth. You're adding one cell at a time when you divide, whereas when you have every cell dividing, you get exponential growth. And that's something that is worth thinking about. In real tissues, having every cell in the tissue divide simultaneously is unusual. It happens early in development and not typically in other tissues. And then we explored last time, both in class and in the homework, this idea of using pressure to control cell growth. And looking at the homework assignments, there were a few issues that came up. One of them was the fact that the pressure as returned by CompuCell is the negative of what you expect it to be. So if a cell is being compressed, the pressure is negative rather than positive. And so there's a minus sign at issue. And then when you're using a functional form that looks like Pref over Pref plus P, or Pref over Pref minus P, you have the possibility that if P changes sign, this could go, the denominator could go to 0 and your growth rate could go to infinity. And so if you use a functional form like that, you have to do something to make sure that that doesn't happen. So you say, if the pressure is positive, then I do some kind of suppression. But if the pressure is negative, then I just use a value of 1. So I have to have some kind of cutoff in my model. If you use a Hill function and the exponent is an integer, the exponent is an even number. Doesn't have to be an integer, but if it's an even integer, then you don't have to worry about the sign of the pressure. But some people used a minus sign here in the Hill function. And then again, you can have Pref equals P, and then this is 0 and it blows up. So people did, I think, quite a nice job of exploring some of the consequences of things like contact inhibition of proliferation. It's a big topic, and I didn't expect people to do everything with it. But it's something to develop an intuition about, keep playing with it. I understand that writing it up formally for the homework takes time, but running it on your computer and playing with it doesn't take that much time. So I encourage you to take those simulations that you develop yourself and keep playing with them to build up your feelings about how cells behave. And we looked at some of the different cases. For these 3 cases, what happens to the average pressure, for example, in the tissue. In these 3 cases, some people had Well, yes, a non-integer Hill n can run into complex numbers if the pressure is not positive. Yes, true. So you don't want to be taking the -1 to the 1/2. Yes, good point. In general, using a non-integer exponent here doesn't give you much, but it's technically it's not a requirement. And I had asked people to tell me whether the proliferation stopped or not for these three cases, and whether the pressure reached a steady state or whether it kept going up. And people got somewhat different results. Some of that depends on whether cells are dividing at a target volume or target. It's hard. I have to be careful. If cells divide when they reach a fixed target volume or when they reach a fixed volume. Otherwise, I was going to say target volume, which is in target volume, but not target volume. So there's some ambiguity in the terminology. And it depends also a little bit on the parameters you pick, on the compressibility of the cells, on the growth rate. And these are things that you also see to some extent in real tissues. Again, I'm not going to go into all of the possibilities, but people explored that. And then there was a homework problem I gave you where you had differential adhesion and cell division happening at the same time. And people saw that you could get all sorts of different patterns when you combine cell division and adhesion. And a lot of those patterning mechanisms are present in development in one tissue or another. Almost every one that we explored in that homework problem exists at some point in some organism. And then I want to now come back to the main focus of today's lecture, which was concentration fields. Now, we started out by the idea of what is a concentration field, and we're pretty used to saying that the concentration of sugar in our solution is so many moles. But we do have to remember that when we're talking about chemical, the chemical is in fact discrete. It comes in molecules. And our ability to talk about a concentration depends on there being some volume in space that has enough molecules in it that there is a relatively continuous variation in the density of molecules per box as we move in space. And the boxes are also small enough that we have some meaningful spatial variation over the system we're looking at. And I'll point out that in this picture here, this is not true. The boxes that are drawn in this picture are so small that you have one particle in them, 2 particles, 3 particles in them. It's not approaching a continuum. And so actually the existence of this mesoscopic scale that makes fields a reasonable approximation is not obvious in biology. And I gave you the example of DNA, where you might have a few molecules of DNA. And to talk about the concentration of DNA in a cell in so many picomolar makes no sense. Many smaller molecules also exist in very small copy numbers. In particular, transcription factors often occur 3 per cell, 5 per cell. And to give them a concentration rather than represent them individually doesn't really make a lot of sense. Rather surprisingly, assuming things behave as concentrations often gives you a reasonable approximation to the biological behavior, even though it's mathematically and physically unjustified. And the main reason for that is that biological systems are extremely noisy, and so they also have to be very robust to approximations. If biological systems were only as robust as human-engineered systems are, none of our models would work, because our models are such coarse approximations that we depend on the fact that you don't have to get the details right for things to work. In our case, we're going to represent fields, whether chemicals or something else, on a lattice, and there'll be a real number at each position in the lattice, which represents the amount of the, or concentration of the chemical. In CompuCell, the lattice discretization of the field is the same as the lattice discretization of the cells. And that has some awkwardness. It would be actually nicer if CompuCell would let you have a lattice that had half as many voxels or twice as many voxels as the cell field, or that let you have more density of voxels where the concentration was changing rapidly and fewer far away. At least being able to do more or fewer by a factor of 2 should actually be possible. It wouldn't be hard. That's something maybe should be an extension of CompuCell. We talked a little bit about diffusion. The random movement of molecules leads to a general flow of concentration from high concentrations to low concentrations. If I draw a line in the middle of this box, and I say that each particle is equally likely to move to the left and the right, then even though the individual particles don't have a bias in their movement, the net flux will have a bias because the number of particles on my left is bigger than the number of particles on my right. And so the fraction of particles moving to the right is larger than the number moving to the left, even though the individual particles aren't biased. And that is physics section entropic argument if you're a physicist. And it leads to the gradual uniformity of the concentration. And we'll talk quite a bit as we go through this lecture on diffusion and its role as a function of length. And something that we're going to say again and again is that diffusion is mostly important over short distances. Diffusion is a very effective means of getting chemicals or heat or charge or electrical field or force over short lengths, few cell diameters, microns, millimeters. It's not an effective way to get molecules or anything else over long distances. And this is something that we experience if you have a radiator in the corner of your room. It'll heat up the area around it. If you put a fan to mix the air in your room, it will work much more effectively. It'll distribute the heat much more effectively. In the case of heat, you also have radiative transport as well as advective and thermal conductive transport. But the basic idea is the similar. And then we talked just a little bit about the fact that biological systems are in fact crowded, and that means we don't typically have what's called free diffusion, like sugar molecules in a glass of water. Rather, our molecules have to move around obstacles. And if nothing else, that means that the distance they have to travel to get from one end of an object to another is longer than you would think. And so that slows down diffusive transport. In some cases, it can do more complicated things like change the exponents or the temporal properties of transport in complicated ways. And there's a whole physics of what's called anomalous diffusion, where diffusion is not quite what you expect. The other thing that we face in biology, in which CompuCell is not tremendously good at, and I was just looking when I was pulling the papers of Stan Murray, his most highly cited paper is on root development in plants. And root development in plants is governed by a signaling molecule called auxin. And auxin has the property that it is transported across plant root membranes by an active transporter that pumps it unidirectionally. And so modeling oxygen actually requires modeling transport, unidirectional transport across membranes. That's something that's rather hard to do in CompuCell. It's not impossible, but it's not very convenient. When you want a molecule to get across a membrane, you can have direct diffusion across the membrane. That works very well for things like water, small ions, and small molecules like glucose, ATP. But it doesn't work so well for large molecules. It depends also on the hydrophobicity or hydrophilicity of the molecule. You also have ion channels, which are essentially holes in the membrane, and the diameter of the hole will control what size molecules can go through it. Both of those are things that we can model relatively easily. More complicated is what here is called facilitated diffusion, where you have active pumping of one sort or another. And we talked a little bit about all the different kinds of pumps that exist. And we're not going to have any pre-built utilities to do pumps. We have to build those by hand if we need them. We talked about how to derive the diffusion equation. I skipped this a little bit. I'll leave it in the slides. If you're interested, you could look it up in Wikipedia. They have a good article on it. The key idea is you define a concentration. Here, the number of particles within a box. Here, sides x, y, and z, delta x, delta y, delta z. And we ask, what is the transport due to this random movement of the particles? And effectively, what we see is crossing a boundary. The rate of transport from the left to the right is proportional to the number of particles on the left, because the random movement is uniform. The rate of transport backwards from the right to the left is proportional to the number of particles on the right. And so the net transport is the difference between the number of particles on the left compared to the number of particles on the right. That is precisely the gradient, the slope of the concentration. And so the flux across a boundary is proportional to the slope of the concentration. And it's a gradient, a vector quantity, because you have a slope in the x direction, y direction, z direction. If I draw a little cube, I see that I can treat these as independent. The direct slope in the x direction, the y direction, the z direction can be handled separately. And so you actually have a vector, which is the flux through this object, which is the gradient in each direction separately. So if I don't like vector calculus, I can do it in each direction one at a time, and it's regular calculus rather than vector calculus. D here represents effectively the typical speed of the particles. The faster the particles are moving, the faster the thing equilibrates. That gives me the flux. Now I want to know what the change in time is over my box. So I have to basically then ask the question how this flux changes. The net flux over the whole thing is the flux over each side summed up, and that's given by the divergence, and that gives me the change in concentration per time. If the diffusion constant is uniform in space in all directions, and it's the same everywhere, then I can move the diffusion constant outside of the derivative, and I get the classic diffusion equation, d times del squared c. In biology, very often the diffusion equation is not uniform in direction, and it varies in space. And therefore, this form that we see here is the technically correct one. And I'd actually have to write it as a tensor if I wanted to do it right, which I'm not going to do. We're going to live with the approximation here for the most part. We also talked about boundary conditions. We have to know what happens at the edges of our box. The simplest thing would be to assume that there are no chemicals moving outside of the boundary. And if I were in a test tube, the chemicals in my test tube shouldn't be crossing the glass, at least if I've picked the glass properly. But cell boundaries typically are permeable, and usually we're not representing the whole test tube, we're representing some finite volume of space where there is no hard boundary. And so our molecules should be able to cross the boundary. And typically what we'll do is allow them to come back on the other side when we do that. We'll use periodic boundary conditions. We'll also find that it's very... Josh, question.
Well, I just had a quick question about, there was a homework problem related to this. I don't know if I did it incorrectly but you said it was periodic boundary conditions and then an X and Y, and then an initial concentration of 1 everywhere. And when I ran that, there was nothing really happened, like no diffusion. It was just constant concentration everywhere, and it didn't change. Is that the expected behavior, or did
Yes.
That's the expected behavior?
I think I actually wrote that in my comment on your homework, that was precisely what you expect.
Okay.
If so, let's come back to this diffusion equation. This is saying that if the concentration is the same everywhere, the gradient is 0. So there is no flux, so there is no movement of chemical, net movement of chemical. So if the concentration is constant everywhere, del squared, the Laplacian of C is 0, and so the change of C with time is 0. So diffusion by itself always acts to flatten concentrations. It acts to make the concentration uniform. If the concentration is already spatially uniform, it doesn't do anything.
Okay.
Now, if the concentration is uniform within a region, and you impose boundary conditions on it that are non-uniform, then that will do something else. And that's what we'll do in class as soon as we get to past the next few slides.
Okay.
But yes, the first exercise was start with a concentration of 1 everywhere, boundary of 1 everywhere, or periodic boundary conditions, and nothing happens. So you got exactly the right answer.
Okay. And then I don't know if you'll talk about this later, but one thing that you tell us to think carefully about whether we use fixed or auto-scaled color ranges, since these will show different things about the field. I haven't been able to find any settings about the color ranges, so I don't know what I'm using or how to change it.
Okay, we'll go over that in a few minutes.
Okay. Thank you.
And if there's no documentation about that, Giuliano, that when you describe how to use player, we need to document that. I realized also about making movies. We need to have better documentation about how to make movies. I was going to ask, I'm trying to remember who did the nice movies of the bacterium macrophage simulation. Was it Joel? Somebody had nice movies in their homework. I was going to say maybe I could have you, whoever it was, demo the movie making so that people all learn how to do that. So whoever it was, you could be prepared for later in the class doing a demo of that. Good question. Okay. So then we did exactly the thing that you were asking about, which turned into a homework assignment, which was create a simulation with a single field, diffusion constant set to 1, no decay, uniform concentration 1, periodic boundary conditions, and see what happens. The answer is nothing happens. Now change constant value boundary conditions. If the value is 1, nothing happens. If the value is 0, things start happening. If you have constant derivative boundary conditions, you'll see things happen. And so the idea here was you start with a situation where nothing happens, and then you see that changing the boundary conditions changes the result. And so the point of that exercise was essentially to emphasize how important it is to think about the boundary conditions when you're doing diffusion. In the case of cells, you can build a situation where there's medium around your cells, and so the cells never bump into the boundary of the simulation. And in that case, you don't have to worry so much about boundary conditions. When you have diffusion, you inevitably have to decide what the boundary conditions are. You can't get away with not thinking about it. And so that's something that, again, if you come from a physics background or chemistry background, diffusion is something you learn about. If you haven't experienced that, then building up the intuition about it may take a little bit of effort. Okay. And then I had another problem where we said, okay, we're going to build a concentration where there's a peak in the middle and see what happens. That would be one we could play with here. And then here I also did one which was affected by the distance to the boundary. And how many people did all of these examples? Nobody? Some?
I
Would it
did
be
like
useful?
2 of those. Yeah,
Okay.
but
Would it be useful to do these again in class as an exercise, or shall we go on? You tell me. I'm happy to review it with you and do it as an exercise in class if it's helpful, but I don't want to take the time if it's not, so you have to tell me what's useful.
I definitely messed something up in mine, but if no one else messed up theirs, then I'm happy to just continue on my own.
Well, why don't we do it? Why don't we do it? If you're bored, you can try playing with things. There's a long list here of possible things to do. And if you get finished through that list, you can invent things to do. I promise you that even this very simple case, you could spend several hours exploring it and not explore everything that's possible. So let's make this into an exercise. Okay, so what I'd like you to do is start out by creating a simulation with one field, diffusion coefficient set to 1, no decay, uniform initial concentration, periodic boundary conditions, and see what happens. And we already know from Josh's, the answer is that nothing will happen, but let's make sure nothing happens. And let's all get to that together. And then once we've got that together, we can go forward and play. And again, one of the whole points of CompuCell is to let you play. And I realize that when you're learning it, it may seem like play is not the game, not what you're doing. This is not fun. But in fact, I would say that the kinds of things we're doing are like learning a language. Learning grammar and vocabulary is not fun. Speaking, joking, reading is fun. And so there's a certain amount of scut work that is required to build the vocabulary and the grammar. But then you can play. And I hope by the end of the semester, everyone is at the point where they can play and enjoy building simulations, as well as thinking, I have to build simulations. So why don't we all take a few minutes to do this? I will do it in conjunction with you. But please do it on your own computer. And I will do it silently at the same time, just so that I can do it. I don't have it pre-done, but I'll do it too. And Giuliano may be the same thing. Okay. If there are any questions, we can please feel free to ask. If you were watching me type, which I asked you not to do, you will see I made plenty of typos while I was doing that. Everybody's ahead of me. You did it faster than I did. Okay, so here's my solution. I get 1. Not very exciting. Nothing's happened. So now let me go in here. That's interesting, Giuliano. Did you notice this?
What?
I have no cells,
Yeah.
and I cannot pause the simulation.
Huh.
I think there must be some rule that says that if there are no cells in the simulation, it doesn't check the pause button.
Wait, is it? I
can
don't think it's
verify.
running right now.
Is it stop?
Yeah, there.
Okay, why did it? Okay, it stopped itself. I don't actually know what happened. All right, fine. Thank you. I can be wrong in all sorts of ways. Okay. So let's
And
come back now. So go ahead.
No, I was gonna say I think the cell thing was just dependent on however you end up commenting out the cell initialization. Because I think I didn't see how you did it, but I commented out the whole block and I don't have it saying if there's any cells there.
Okay. That's fine. I turned off the cell. Yeah, I did the same thing, commenting out the block. One thing that has been fixed in the current version of CompuCell is it used to be that if you commented a comment in XML, it crashed. Because it winds up that if you have the uncomment command twice, it causes a problem. And so now the block comment is smart, and so it looks and sees it. If there's a comment inside of the thing you're commenting, it keeps it as a comment. That was actually work. I don't know how they came up with this strange comment syntax in XML, but putting a comment inside of a comment is illegal syntax in XML. Gives you a mismatched tag. So that now works correctly. So okay, so this was periodic boundary conditions. Let's do constant value boundary conditions. Somebody, why don't you tell me what happens when you go to constant value boundary conditions?
You have basically two, one max and one min on top and one on the bottom.
So if I make the values 1 and 1, I'm just going to do it on the top and the bottom here. If I make the values 1 and 1, and now I come back and do it again, nothing happens. Josh, go ahead.
Yeah, sorry, I was just a little confused at the wording in the homework. It's a similar problem. I think it's the exact same one actually, but you say change the boundary conditions to be a constant value at both the min and max of the X and Y directions, set the boundary values to 1. Does that mean set min and max of X and Y all to 1, or just any constant values?
No, so here you have constant value,
Mm-hmm.
min, X per direction, constant value, minimum and max. Because you have two boundaries. You have the boundary on the left and the boundary on the right.
Mm-hmm.
And then if you're setting a constant value, you have to say what the value is. And so let me do that. Here I've set it to 1. And let me do it for the Y as well. I was going to just do X, but it's fine to do Y too. We'll do Y as well. And notice that the default here is actually constant derivative because I want to illustrate different ones. I have to change that to constant value. And you have to comment out the periodic. If you have both of them, it'll get confused. I don't think it'll crash. I think it's just whichever one is later in the tree will work. And here I have to set the values, so I'll set the values to 1. And let's see if that works. If this worked, I'll have nothing. Sure enough, nothing. Now, suppose that I go in, and I can do this in the model editor here, so I don't have to restart everything. I go into my boundary conditions, and I change this value from 1 to 2. What's going to happen? Somebody. What'll happen?
Does that just mean that there's going to be a higher concentration on the x-axis than on the y-axis? So there will be diffusion?
In or out?
So it always goes from high concentration to low concentration. So it'll be going from X to Y.
Let's try. I've created a source on the left and the right. You see the chemical flowing in.
Mhm.
I didn't change the top and the bottom. Notice here that the concentration is going down as I near the top and the bottom. That's because those are fixed at 1. If I had periodic boundary conditions on the other direction, you'd see things moving in without that curvature at the top and the bottom. That's why I'm saying there's a lot to explore even in this very, very simple exercise. So this is, you see it flow in from the edge and flow out at the top and the bottom. If I now were to go in, let me do this. If I now went in and did this, now I'll do it permanently, so to speak. I'll make the top and the bottom 2. But I did that's the left and the right. Let me do the top and sorry, the top, left and the right. I'll make these 2, which is what I just did. And now I'm going to go back to being periodic. Unfortunately, I can't change the boundary conditions on the fly in the simulation. I have to stop it and restart. So now look, remember what that looks like. I suppose I could take a screenshot of it, right? You can take a screenshot. Look at this curved lines that you're seeing here. I'm now going to run the same thing with periodic boundary conditions. And now you'll see moving in without that curvature at the top and the bottom. So that's the interaction of the top boundary conditions with the left-right boundary conditions. What if instead of making the values at the boundary bigger than 1, I made them less than 1? I can do that in the model editor. I could make this now 1/2. Let's make it 0. Now it's a little bit harder to see, but I'm actually having chemical flowing out the other way, coming out. Notice that the maximum value is still 1 here in the middle, but the value at the boundary is now 0. So I have flow out. What if I made it uneven? What if I had the input side on the left-hand side was 2, and the right-hand side was 0? Two here, 0 here. I'm getting something that's doing this. And as time goes on, it'll give me a gradient. So you see, even this very, very simple case, I actually have rather a lot that I can have happen. Constant derivative boundary conditions are a little bit more complicated to think about. They basically correspond to either forcing the fluid in or chemical in or pulling it out. And a negative constant derivative is a little problematic because you could be pulling something out when there isn't any. You can get a negative value, which you're not supposed to get. Do you want to try exploring that a little bit yourselves? Why don't you try playing with it? Just take a few minutes and try some of these breaks. Do you see how to make the changes? If you want to change whether it's constant value, constant derivative, or periodic, I'm afraid you have to go to Twit and change it here. If once you've got it set up, you can change the values by hand in the model editor, so you don't have to restart. Constant derivative was, remember, we had that before, constant derivative, just the word derivative. Constant derivative is also known as constant flux. So why doesn't everybody just take a minute or two to try that out? And if you find something interesting, a pattern that looks interesting to you, you could show us and tell us what conditions gave it to us. Okay?
I have one that I don't think is biologically realistic at all necessarily, but it's fun to look at.
Let's take a look.
So I went back to constant values and then I set it so all of them were 0 except for 1, just because I wanted to watch it play out, and it's kind of fun. I think y'all can see it right now. Let me know if share screen isn't working. Sometimes I still mess that up, even though Zoom has been okay. So yeah, I'm gonna eventually.
Okay, so let me just, well, there was a question about, and I have some slides on it, but let's do this while you've got it screen shared.
Okay.
Can you open up the settings menu, the settings menu? Up there.
Right here. Oh, yes. You want to go to this?
Field, yeah. And so you have the opportunity of setting constant minimum and maximum values, or float the top or the bottom value. And then you also have gradients, didn't you? You have You have
I
contours here on the right.
yeah.
Scalar contours. And if you let's leave those on, and you can point out what they are in the display. So
Okay, so go ahead and
say okay, so hit keep running, and then
yeah. So now the min, so now it's, before there was slight fluctuation at the min and max, but now it's keeping the scale of 0 to 1, and these are the contours that I turned on right here.
I think it was Josh who asked about this, right? That we hadn't shown how to do that. So does that help out with the things you were worrying about?
Yeah, I found that. I just didn't know that that was the color bar range, the min and max. But yeah, all right, I understand now. Thank you.
Okay. No, I mean, as I say, that there's so many things to cover that it's very helpful to have a question like that. I'm sure there are lots of people who would have the same question. So please keep asking those questions. I appreciate it. Okay, does anybody else have something they'd like to show us? Oops. What am I screen sharing?
I can show, oh, you're showing everything, yeah. That
I'm
you were
showing.
showing previously.
Am I showing Zoom twice? Are you seeing yourself?
No.
Okay, because sometimes it'll share the video feed. So
No, we are
then you
seeing
get you
the
can't find.
PowerPoint and NanoHub.
I can show you a bug if you want to see one.
Okay, if you like.
Yeah, the stop button is not working. Look, it's not pausing. Remember that thing? It's still running. I think I showed you that before, right, Giuliano, or something? Yeah, but I couldn't ever got in record or anything. So here it is.
Yeah, I don't
Yeah.
remember
So
you showing me that, but I have seen it over here. And
right.
And when I press stop, then it crashes. Yeah, that's the thing. Sorry.
So don't, before you kill that, I want to point something out, which is you have a concentration that's negative, so you must have constant flux boundary, constant derivative boundary conditions.
Yes. Mm-hmm.
So the diffusion equation, in principle, chemical concentrations can't go negative. The diffusion equation doesn't care whether values are positive or negative, so it's well behaved for negative values. It just isn't meaningful for concentration. For electrical charge, you could have positive and negative electrical charge, so you can have a negative value in diffusion. So here you have negative 104 is blue.
Yep.
Okay. Anybody else have anything that they explored with that, or is it?
No, I can say that the negative values were what convinced me that I was doing something wrong in the homework originally, because I was messing with the oxygen field, and the moment it told me that I had negative 200 oxygen, I felt like something was off.
So one thing that you have to be aware of when you do constant derivative boundary conditions is that the derivative is with respect to the x-axis. And so a constant derivative on the left, if you have a constant derivative on the left that's positive, it takes material out. A constant derivative on the right that's positive puts material in. And so to have the things to be symmetrical, on the left and the right, you have to have minus on one end and plus on the other, because the direction is opposite with respect to the axis. And so that's a little bit of a minefield there for people, because you think minus one should do the same thing on either end. But because you're relative to the axis, not relative to the center and inside and outside, you have to flip the sign. The only other thing I think might be worthwhile is trying something like this, which is, did anybody try this number 2 where you set an initial concentration peak in the middle? So the initial concentration here, you can't do very fancy things with the initial concentration, but you can do simple algebra. And a few functions like max and min, sine and cosine work. And so I can do max of 0, comma, let's try this, 10 minus, you need a lot of parentheses, x minus 100 squared, and unfortunately the squared caret, the caret is, here is caret rather than star star, so it's not Python. Squared plus parenthesis y minus 100 quantity squared. See if I got my parentheses right. What will that do? See if it crashes. Oops. Oh. Let me go back to periodic boundary conditions. And again, if I'm playing like this, I hope you play too. Please feel free to play any way you like. It's plenty to explore. Well, I crashed CompuCell. I don't know how I managed to do that. Let's see what happens. Did anybody get that to work?
I think I did, but I did what you suggested in the homework where you just select the field through the self.field.oxygen and then just set it equal to 1 based on the coordinates.
So you did it in Python.
Yeah.
Well, that should, I mean, Python should work. Let's see. Here I have diffusion from the corners here, but it says 5 everywhere, which is sort of odd. Let me see what happened. Field is floating everywhere, so it should be all right. So this is actually, let me open Twitter. Did anybody else get this to work?
I got a different result, yeah. I don't know if this correct result that you're looking.
I think I did something. I clearly made a mess of the initial concentration somehow. Max of x minus 100 squared. Well, what size is my what size is my simulation? I probably was trying to make it be, oh, it's 256, so I should probably have made this 200. You want to show us? I don't want to take too much time with it, but you can play with it. The idea
Sure.
here was to make a pulse in the middle or a pulse at the edge. Let's see what happens. Good.
It's actually I kept it running. Let's see if I can pause it.
That's fine. That looks great. That's what I was hoping to have. Yes.
just let me know whenever you want it back, the sharing.
No, that's fine. Can you show the code too that generated that?
Yes, yes, it's right here.
Maybe zoom in a little bit so people can see. So it does work.
Yeah, it's
I
this
must
one.
have had a parenthesis off when I typed.
Yeah, and I think I turned off every. Oh, I put periodic. Yeah,
Right.
periodic here, periodic
Good.
here.
Thank you. Thank you. Let me get the screen share back. All right. So let me just do the analysis very quickly of this, and then we'll take a break in just a few minutes. So let's think about whether we can do analytically. We haven't done much math in this class, and I try to minimize the amount of math in this class. I know some people like math, but some people who take this class aren't so fond of it, so I try to minimize the amount of math you have to do. If you like math, I promise you can have as much as you want. So we're going to say, well, what if we just fix the left and the right of the simulation? We did the example of constant value to the left, constant value to the right. And imagine periodic boundary conditions top and bottom. If we have periodic top and bottom, basically the y direction isn't doing anything. So we basically have a one-dimensional simulation. In that case, the derivative with respect to time is d, and now instead of having a Laplacian, we simply have the second derivative of the concentration with respect to x. And now we ask, what is the equilibrium solution? What's the time-independent solution? To do that, we say dc by dt is 0. So we set the left hand of the side of this equation to 0. D times dc by dt dx quantity squared. So the second derivative of c with respect to x times d is 0. That means, in particular, that the d goes away, it doesn't matter, that the derivative of c with respect to x is a constant. That's the first integral of 0. And that means c of x is a constant times x plus a constant. So that means our concentration is a straight line. And we have to pick the values of the constants so that at the left-hand side it has the right value, and the right-hand side it has the right value. So we get a straight line. It's what's called the linear gradient. So the profile is linear and independent of the diffusion constant. If we set that it's a value of 100 on the left and a value of 0 on the right, and our box size is 100, then c of x is 100 minus x. If x is 0, it's 100. If x is 100, it's 0. Now, the length of time our chemical takes to diffuse is going to depend on the diffusion constant, but the equilibrium is independent. And we'll come back to that length of time question in a minute. Does that make sense? So now I want you to do a short exercise, and then we'll take a break after the exercise. I want you to write a simulation where we impose a constant concentration on the left, constant concentration on the right, periodic boundary conditions on top and bottom. We just did that. And the next thing that I would ask you to do is to plot a graph where you go across the field from the left to the right, measure the concentration along the middle, and plot the concentration versus position on the y-axis. I think you had that in the homework, so you should have it available to you. If not, you may have to ask the question, one, how do you get a reference to a chemical field? and then you have to iterate over a position. So is the exercise clear, the way it's defined? Okay, so why don't people work on that for a few minutes? That may take maybe more than 1 or 2 minutes. It might take 5 minutes to do. Giuliano, you look puzzled. Is there something wrong with the way I defined it? Looks okay. So why don't we do that exercise? And then we'll come back. We'll do our break in the exercise together. So we'll come back at 7:05. And my hope is that somebody will have done this so they can show it off to us. Is that okay?
Yeah, can you just show before we go how to get the chemical field? Because I was having trouble with actually finding. There is a definition of like value equal field, and then it gives me a vector. But the vector is already defined. It's not like asking to get the vector on Python.
So let's go to the Python. And so we'll go into our step function here. We'll get rid of this stuff. And we'll go to CC3D Python, and we'll go chemical field manipulation. And we'll go to get field reference. And it'll say field equals self dot field. You have to type here the field name. And in my case, I called it oxygen. And the trick is you have to put it in all caps. Is that right still, Giuliano? Or has it changed?
I think the field you write it exactly the same way as in the
Okay.
XML.
It used to be different. All right, so you have to check oxygen. Okay. And then we can iterate, and then we can get a value reference. So we can say chemical field manipulation, get, let's see, get field value.
Yeah, that's the one. It gives the 10, and that's the vector that I have to pass as the x and y values that I want. Max. Oh, I see. Makes
For
sense.
x in range 0, comma, x. I always get it wrong. Dimension self.dim xdim. What's the
Self.dim.x or y.
and then I'd add the point to the. I can do it if you like, but I think I'd like people to try it on your own. Oops. Everybody's watching me. You should be doing it yourself.
So I actually had a quick question because I had to minimize while, one computer screen. So I got partway there, but I'm still lost where Joel was lost, which is that I think I got partway through what I needed to do for referencing along the x-axis, but something is wrong with my code.
Okay, let me walk you through it then. Did everybody try it? Did other people have problems? Did anybody else have problems? Let me expand this a little bit so we can see.
Yeah, one of the problems that what I was having was the oxygen name was giving me the oxygen was not defined or something.
So you have
That's solved.
to get a reference to the field. So here I say field equals self dot field, and it has to be the name you used. And I was wrong about saying it was capitalized. That was old world. So it has to be what you did. Once you have that reference, then I can read the value of my field at any position. So here I'm iterating from 0 to the size of the lattice. I read the value at each position. I'm going to pick Y position of 10. Could be anywhere because it's the same. Probably if I wanted to be a little more formal, I would use, if I wanted to be in the middle, I would type self dot dim dot Y divided by 2 to give me the middle line. Okay. And now I add a data point in my concentration, which has the X position and the value. Now, one thing that this will do is it'll keep overwriting. So each time it adds to the plot rather than updating it. So I probably don't want to do this. I probably don't want to do this every Monte Carlo step because it's going to plot too much. And so I would probably do something like if, and I know it's not very elegant, if not MCS %10, colon, that'll do things every 10 Monte Carlo steps. The other thing I could do would be to do something which we haven't done much yet, so far, which is you might erase your plot. So you only have the latest one. Does that make sense now to people? Does everybody want to look at the code and try it out and make sure it works for them? And again, your name may vary, whatever the name of the field you used was. Okay, so you got the whole creating the plot window, adding the plot, everything here. So I'm going to take my 5-minute break and I'll come back at 7:10. We'll keep going. Oops. Okay, people back? Great. So people able to catch up on that one and get that to work now? Does anybody need help still? Feel free to speak up. So just to remind you, and I think I can skip this now. I have a walkthrough of all of the lines in this. Again, the key things that we're going to do is set the diffusion constant, decay constant to 0. We'll set the minimum maximum values to 10 from 10 to 0. And the y-axis we set to periodic boundary conditions. And then we have to decide how we're going to visualize the field, which we talked about already. And in this case, we could use a fixed range. For example, minimum is 10 and minimum is 0, maximum is 10. We could show the legends, show the contours, and so on. So we have the choice of fixed or floating boundary conditions, top and bottom value of the color map. And if we wanted to save a movie of this, we could go into the output menu. And under output, we could tell it to save the concentration field every so often, if we like. And then remember something I think everybody's figured out, but I have a choice here in my display. I could display the cells, of which there aren't any in this case, or I could display my chemical concentration. So if it's set for default of displaying cells, I have to switch the display. And indeed, it looks pretty much as advertised here. Notice that I said that the equilibrium value would be a straight line, but even though it's been running quite a while, I still don't have a straight line. And one of the things that we could do, which is what we've done here, is measuring the field. Again, here the diffusion field name was oxygen. We get the concentration field for oxygen. Then we use the value field, iterate over the range, and add the data point. As I mentioned here, plotting all the data points takes a lot of time, so you might want to erase the field this graph periodically. Let's see what happens if we do that. That'll only take a second. So let me do an erase here. We haven't done that yet. CC3D Python, scientific plot, erase plot. Not so pretty, it's blinking a bit. And now you could see the line moving up gradually as the chemical diffuses. I think if you do this on your screens, it should be smoother. I'm actually surprised that it's blinking. I think that...
Isn't it blinking because you put it on every MCS? Maybe?
Did I do the delete in the M outside of, oh yeah, sorry. My mistake. Thank you. Let's see what we get now. Yeah, thank you. That's it. I was erasing it 10 times for one grow. So now you get a pretty smooth movement. Depending on what you want, you might want to see the whole history. So you might want to leave them up. But here, if you want to see how it's evolving, it's easy. You'll notice it's taking a long time for that chemical to get from the left to the right. And so that's the next thing I want to talk about. Is again, we start out with nothing. The chemical begins to diffuse from the left. After 120 Monte Carlo steps, the chemical has moved about 10 voxels in. We can try that. Let's try this by step it. Start it again. I start with nothing. I do 1 Monte Carlo step. I get a little bit of chemical on the edge. I go to 10, and it's beginning to come in a little bit. You could ask the question, how far has it gotten as I run? Let's run to about 100. Here I'm at 200. Square root of 200 is about 15. I'm at about 15 here. If I let it run more, let's get to 900. It runs fast, so I don't have to worry about that too much. 900, if I take 1 over E, I've gotten to about 30. If I wanted to get all the way across to 200, I can tell you what the answer is. I'd have to go to 40,000 time steps. To get this to smooth out, it would take 40,000 time steps. In this one here, I've made a smaller plot. This is 100 by 100 instead of 200 by 200. An example I have on that. And this is going to bring up the concept of a diffusion time. And the diffusion time is the time it takes a molecule to go a typical distance. And if I'm driving a car at 50 miles an hour, then the time it takes to go 50 miles is an hour. The time it takes to go 100 miles is 2 hours. So the time goes linearly in the distance. With diffusion, the time goes like the square of the distance divided by the diffusion constant. In this case, my diffusion constant was set to 1. And so the time is the square of the distance. In this case, to get to 200 is going to take on the order of 40,000 Monte Carlo steps. The value of the diffusion constant in biology depends enormously on how big the molecule is and what it is. And really, a lot. And this is a problem when you're modeling, because dealing with different time scales in the same model is difficult. In our world, typically, if we have a very small ion like potassium or sodium in water, this diffuses 2,000 microns squared per second. And we're going to assume that that diffusion is so fast compared to most of the biological processes we care about that it's instant. And then we'd use the steady state diffusion solver. Somebody used the steady state diffusion solver in one of their homework problems. We tend to avoid it, but that's when you need it. What do these times mean? We're going to have to think about what these diffusion times mean, the constants mean in a minute. For a molecule that experimentalists use a lot, green fluorescent protein, and now inside of an E. coli rather than in water, the diffusion constant is about 7 microns squared per second. And that's going to be fairly typical for small molecules. DNA is big, and here we're in yeast, so the chromosomes are not huge, but are pretty big. The diffusion constant is 5 × 10⁻⁴ microns squared per second. So I have a difference here of 10⁷ in diffusion constants between my ion and the DNA. Now you might ask, what determines the diffusion constant and how do I estimate it? That's a little bit tricky. There is no simple way to calculate the diffusion constant for a given molecule. There is a relationship called the Stokes-Einstein relation that says that if you have a spherical object and you're in a simple fluid like water, that the diffusion constant of that sphere decreases with the radius of the sphere, decreases with the diffusion constant, decreases with the viscosity of the liquid, and increases with the temperature. And that particular relationship sets an upper bound on the possible diffusion constant for a molecule. In reality, biological proteins aren't spheres. They're a little bit extended. And so the diffusion constant will be slower than you would guess if you had a sphere of the mass of the molecule that you're thinking of. And the fluids themselves are not perfect, and that also will slow things down. But typically for most molecules, the actual diffusion constant is within an order of magnitude of the Stokes-Einstein estimate. It'll be less. Exactly how much less you have to do experimentally. If you're a chemist, you can get some estimates, rough estimates of how much less, but there is no analytic way of determining. But let's take just the example we have. For oxygen, I should have really used oxygen instead of an ion here. The diffusion constant for oxygen is on the comparable to that here. It's a little less. It's about 2,000 microns squared per second. So we want to know how long is it going to take for oxygen to go 100 microns. 100 squared is 10,000. 10,000 divided by 1,000 is 10. So to go 100 microns, oxygen takes 10 seconds. To go 1,000 microns, 1 millimeter, how long will it take? Assume D is 1,000, not 10,000. We're going to go 1,000 microns. How long does it take to go 1,000 microns? Everybody's looking at the floor. I'm going to call on somebody. Connor, how long does it take?
My ability to do math off the top of my head is, so wait, so it would just be, You said 1,000, so it's 1,000 squared divided by D, which you said was 2,000.
1,000.
Oh, it's 1,000 in this case. Okay.
I think 1,000 squared divided by
That's what I
1,000
was about to.
was a reasonable question. That's why I
It-
thought it was okay to ask.
No, it is. I always second guess my math, especially when it gets later in the day.
So how long does it take?
It takes 1,000.
1,000 seconds.
Yeah.
So for oxygen to go 1 millimeter takes 1,000 seconds. That's pretty slow. So if I take a blood vessel out of your body, it takes 1,000 seconds for the tissue a millimeter away to know that it's changed.
Also way longer than the 3 to 5 minutes needed for your brain to die.
No. So diffusion works really well over a time scale over a distance of 100 microns. It changes quickly, but for a distance of 1,000 microns, it's slow. Things slow down as the square of the distance. A typical protein, the diffusion constant's 1/10 that of oxygen, 100 microns squared per second. So to go across a single cell has a diameter of 10 microns. 10 microns squared, 100 divided by 100. It takes 1 second for a typical protein to go across a cell. For a typical protein to go a millimeter, 1,000 squared, a million, divided by 100, 10,000 seconds for a typical protein to get 1 millimeter. Very slow. So diffusion cannot work over distances of centimeters. So our bodies can't be built on diffusion. You can't build a biological system based on diffusion that's more than 100 microns to a millimeter thick. And so that means that in bacteria, the biggest bacteria are 100 microns across. Algae are thin. They're 100 microns thick. They can be sheets. They're very long, but their thin dimension is 100 microns, a few hundred microns. If you ask about an axon, if I want to get a molecule from my body up to my brain, because I have a neuron, and I want to do it with diffusion, 1 meter is how many microns? 10 to the 3 times 10 to the 3, 10 to the 6th microns. Squared, 10 to the 12th. 10 to the 12th divided by 100 is 10 to the 10th seconds. That's 300 years. So if neurons operated by diffusion, getting a signal from your foot to your brain would take 300 years. So that's not how those signals are transmitted. So one thing that we can do that is a sort of a fun problem, and I'm debating this because it's a nice problem, but it's a little bit, I'm debating whether I should do this as a homework problem or do it here. What do you think, Juliana?
Do which exercise?
The little membrane example.
I think just showing now would be fun, but
Okay.
doing it now.
It would take most of the rest of the class, wouldn't it?
Yep.
So I talked about membranes and the fact that membranes block the movement of chemicals. And in CompuCell, there isn't a concept of a jump discontinuity at the boundary of a cell. But what you can do is you can build a thin layer of material, which you call a membrane. And you can then ask the question, how does that affect the diffusion of a chemical? And so I could do the same simulation that I've done here, putting a barrier down the middle that was a cellular barrier. And I could change the diffusion constant in that cellular barrier. And then I can, well, let's do it together. I think let's try it together. I think it's reasonable to do it. We'll put a barrier in the middle here and ask what happens to the flow of the chemical, okay? So your exercise is create a simulation of the single field, a single frozen cell type. We're going to draw a membrane down the middle. We're going to set the initial conditions to be exactly what we did here. And we're going to see what happens when we change the diffusion constant. Let me show you two things. In our XML, we have a global diffusion constant here. That's the diffusion constant everywhere. However, we can define a different diffusion constant inside cells of a given type. So this would be, for example, in reality, diffusion of chemicals in water is faster than inside of cells. So we could say that if we're in medium, the diffusion constant is 10 voxel squared per Monte Carlo step. Whereas if we're in cells, the diffusion constant is 1 voxel squared per Monte Carlo step. Does that make sense to people? And so you can try that. To create a initial condition, we should have used uniform initializer instead of blob initializer. So let me get rid of blob initializer here. Put in uniform initializer. And I can draw a layer. I created already a cell type. I didn't do anything with it. Cell type I just called cell. I could have called it wall here. And I can draw a box. And my box could be, for example, box min in the x direction would be 95 to 105. Box max in the y direction, I'm sorry, I did this exactly wrong. 95, and I want y to be 0. I want x max to be 105. And y max to be 200 in my case, because I have 200 by 200 lines. And the other thing that I have to do is something we didn't do until now, which is creating a frozen cell type. And Giuliano, I need you to remind me of what the syntax is to say frozen.
I think it's freeze equals open close quotes, but let me create a frozen cell type real quick.
So you
I
can
also
try
can
this
never
on your
remember.
own, but I'm showing you, and now you'll have to implement it.
Or you can use the CC2DML menu, and it's gonna.
Can it will it
create the-
replace?
It will ask if you want to comment out or replace. Yeah, and it's gonna remember the cells already there.
Cell type, replace.
Yep.
Cell, freeze, add. Okay. Okay. Tells me what I need. Freeze. Okay. Because I don't want my membrane moving in this case. And the last thing I would have to do to make this work is change the diffusion constant in my cell. And I'm going to make the diffusion constant 0 to begin with. What's going to happen when my chemical gets to the wall?
Yes, and could stop there, right?
Well,
And
if I did it right, it should stop, yes. I may have done it wrong, so it may not stop, but looks like it's stopping. So why doesn't everybody try that? Again, this is something it would be good to figure out on your own, but let's try it. I've shown you a little bit about how to do it, so give it a shot. And if you get through that, you can, and you're bored, you can go down this list. One thing we could do by hand, because we can edit it in the model editor, is I can change the diffusion constant by hand inside my cell. If I make the diffusion constant 1, then the cells aren't doing anything, and my chemical crosses the boundary. Now basically I've made a membrane that doesn't obstruct at all. If I make the diffusion constant 0.1, now I get some transmission across the boundary. Look at my concentration. You see I have a kink in it. And I suppose I could have something with an active pump, in which case the diffusion constant at the boundary was actually faster than in the rest of the field. Look what happens to my concentration field. The slope is shallower because I have faster transport. So why don't people take a couple of minutes and try it out? Again, this is one of these exercises where the statement is very simple, but one could spend quite a bit of time exploring the significance. Because one of the things that's important about membranes is that they let some chemicals in these freely and some not. So if you want to do a simulation of calcium waves, for example, you need to know how the calcium interacts with the membrane. If people have any questions about how to do this, feel free to speak up and get some help. But I'll leave the simulation running while you watch. I know our Purdue guests are interested in this particular kind of problem. So maybe it's worth taking a little time. if somebody wants to ask, is more of a physics or mathematics background, you might ask the question, why is this concentration curved rather than straight on the inside? While you're working on that one, I'm going to do number 4. Set that up for you. And I'll leave that for you to do as a homework problem.
Uh, Dr. Glazier, can I actually see the code that you're using to do that? Thank you.
I wouldn't worry about doing number 4 unless you feel like it. I mean, the point was to do number 1, but number 4 is building an ion channel. And if you see what's happened here, I have essentially a uniform concentration of chemical on the left-hand side that's retained by the membrane. I have the chemical leaking through, and I'm getting a hemispherical concentration on the right-hand side. And I have a depletion of the chemical just on the left-hand side of the hole. And so I can see what happens near an ion channel to concentration. But again, the assignment wasn't to do the ion channel, although it's easy enough to do, I think, but just to do the single solid wall. Looks like people are still working on it. You want some more time to work on it? As Giuliano helpfully pointed out, if you forget the syntax, you can go in here, you can select the plugin, go to CC3DML, go to plugin, go to cell type, and then you can do replace, and it gives you the wizard to redo it. So you don't have to remember the command for freeze. It used to be wizard only worked at the beginning of a simulation, but it's been extended so you can use it during pieces of it during the simulation, while you're writing the simulation as an editor. Were people mostly able to get that to work? You were able to get a boundary? If you're able to get the boundary, then the thing to do is to see what happens when you change the diffusion constant across the boundary to make it leaky. That would be to change the diffusion constant of the cell. So here I'm going to make the cell have a little bit of leaking, the membrane be a little bit leaky. Now you see the chemicals beginning to cross the membrane, not only through the channel but elsewhere. And you'll see that the concentration near the membrane is lower than it was in the center. Before it was uniform, now it's lower. And so even this very simple simulation allows you to explore what the concentrations are near membranes that are impermeable, semi-permeable, with ion channels. But the only thing it doesn't let you do is have a selective ion channel. I can't do a pump across that hole. But something like a gap junction or a classical ion channel, I can do. Okay. So I'll set that up as a homework assignment for you. So we've talked about diffusion length a little bit. The next thing that I'm going to talk about is that we have this decay constant. And we've had the decay constant set to 0 so far. And that's assuming that all of the creation and destruction of molecules is happening at our boundaries. And for a molecular species like oxygen or water, we typically are not, those are stable molecular species, although oxygen, of course, is used by cells and converted into other things. However, most proteins and things like RNAs have a finite lifetime. They break down on their own. And therefore, it is pretty common to include a lifetime, a decay time, for our chemical species. We're talking about proteins or DNA, or DNA has a long lifetime, so that's not so much, but RNA has a finite lifetime. Usually, the rate of decay of molecules, and thus something interesting is happening, is proportional to their concentration. The probability of an individual molecule decaying is a constant in time, like radioactive decay. And that's called first-order kinetics. It's also true that if we have no chemical at all, the rate of decay better be 0 because we don't want the concentration to go negative. So that's another reason for linear decay. And with this hypothesis of first-order decay kinetics, that is, the rate of the decay of an individual molecule is constant, we get our diffusion equation has an extra term, which is minus gamma, the decay constant, times the concentration. If we go to one dimension, we get what we had before. The change of concentration per time is the diffusion constant times the second derivative in space minus gamma times the concentration. I can do everything as before, but here I've done a simulation not with CompuCell. I have to say this is from the biophysics textbook that I recommended to you. If I start out with a pulse of a chemical, that pulse will spread due to diffusion and also decrease in amplitude due to decay. The total area under the curve will go down exponentially because of the decay. This gives rise to a concept of diffusion length. Diffusion time is how long does a molecule take to get from one place to another. We did that calculation. Diffusion length is a different question. If I'm likely to be killed on my way, I may never get there. And so the diffusion length asks the question, how far do molecules typically get before they die? And this is also quite important because in addition to the question of how long does it take for something to get somewhere, we have to know whether it gets there at all. And the diffusion length, again, is going to be critical. We're going to see the diffusion length, especially in the context of blood flow. If we have capillaries, capillaries will supply glucose and supply oxygen to tissue. And we need to have a sufficient supply of glucose and oxygen to keep the tissue alive. If those concentrations go too low, the cells couldn't live. And so the separation between capillaries in our body is going to be determined by the diffusion length of oxygen and glucose, how far oxygen and glucose can get into the tissue. We're also going to find later on, when we do patterning, that the diffusion lengths of chemical morphogens, signaling molecules, will determine the size of domains in the early embryo. As the embryo grows and cells replicate, those domains will get stretched, just like drawing a picture on a piece of rubber initially, and then stretching the rubber. But that initial size is going to be determined by diffusion lengths. Diffusion lengths are also going to be very important in adults when you have recovery after damage. During wound healing, diffusion lengths determine how far signals go when you have reconstruction of tissues. Now, there is an out, because while chemicals can only diffuse at a maximum rate, cells can move actively. And so cells can transport information much longer distances than molecules can on their own. The discussion that Connor presented earlier with Drew was asking the question, can a cluster of cells migrate faster than a single cell? And that's actually a rather comparable question. In this case, we're asking the question, can a cell move further effectively than a single molecule? The answer is also yes. And so one of the theories that people like John Lowengrub have developed is that the immune cells in the body are actually sending chemical signals that allow diffusive type patterning to happen over longer length scales. And we'll come back to that a little bit later. It's very important in the context of cancer. If we want to understand the diffusion length, we have to do a little bit of math, and I'll finish this class with this derivation. We're going to assume that we have a time invariant state, like the one we see here, and that means the derivative of the concentration with respect to time is 0. So we set the left-hand side to 0. We now have the second derivative with respect to space, minus gamma c is 0. We now can't eliminate the d anymore the way we did before, and we'll reorganize. We'll put c on the right-hand side, leave the derivative on the left-hand side. Divide through by gamma. Diffusion constant divided by gamma, k constant, times the second derivative with respect to space is equal to my initial value. There are not many differential equations that you have to learn, but one of them is if the derivative of something is itself, what kind of functional form do you have?
Exponential.
Exponential. So as moment you see d by dx of c is c, you have an exponential. Now the fact that it's the second derivative means it could also be a sine or a cosine. And so I'm going to tell you that it's not. If it's a plus sign here, it's not. If there were a minus sign, it would be a sine or a cosine. And so I know that the solution to this is an exponential. And so I write c of x is equal to a e to the b x minus x0, which is the most general form of exponential. I now plug that in here. The second derivative pulls b down twice. So I get a times b squared equals a e to the b. So the second derivative pulls b down twice. D gamma c b squared equals a e to the b x naught. A disappears, b disappears, and I'm left with d over gamma b squared equals 1. I rearrange, put the b on the other side. B equals gamma over d square root. That is, the length is d over gamma square root. And so that tells me that the concentration with decay decays exponentially rather than linearly with distance. And the distance I can get is the square root of d over gamma. If gamma were 0, which is what we started with, the diffusion length is infinite. In this case, the diffusion length is determined by the diffusion constant divided by the decay constant. For most of the molecules we're going to work with, in tissues, the diffusion lengths are typically 5 microns to about 100 microns. And that may be determined by the lifetime of the molecule or the rate at which the tissue takes it up, in the case of oxygen and glucose. And that typical diffusion length in tissues is going to determine all sorts of things about how tissues are structured. So I think we're out of time for today. I'll try to make up now some assignments based on how far we got. I'll challenge you a little bit to go forward with these ideas. And I want to open it up for final questions before we break. If I want to try decay, I could put in a decay constant, a global decay of 0.1. What's going to happen? Why is it not letting me? Oh, because I have 0 here without, it was originally not a float, so I can't make it a float. That's unfortunate. Let's make it a decay constant of 1. Now make the diffusion constant 10. What's going to happen now with the decay constant? Look what's happening. Hence, chemical never gets very far away from the boundary. What is my value? Diffusion constant is 10, decay constant is 1. 10 over 1 is 10, square root is 3. My concentration only gets 3 voxels away from the boundary. Not very far. If I want to get this thing further, I have to make it much smaller, 0.01. It won't let me change it because if it's initially an integer, it has to be an integer. So I could change the diffusion constant to be 100. Now you see it goes further. And so that's something you'll explore. Again, these are not hard calculations, but we have to mentally get used to calculating square roots or squares rather than linear functions. Our brains are very good at saying, if it takes 1 minute to go 1 meter, it takes 2 minutes to go 2 meters. We have to recalibrate mentally to know that it takes not twice as long to go twice as far, but 4 times as long. And the same with the square roots. We have to rethink a little bit about how things scale.